Ammonia transporter. Ammonia transporters TC-1A11 are structurally related membrane transport proteins called AmtB proteins, ammonia transporters in bacteria and plants, methylammonium/ammonium permeases, MEP in yeast, or Rhesus RH proteins in chordates. In humans, the RhAG, RhBG, and RhCG Rhesus proteins constitute solute carrier family 42, whilst RhD and RhCE form the Rh blood group system. The three-dimensional structure of the ammonia transport protein AmtB from Escherichia coli has been determined by X-ray crystallography, revealing a hydrophobic ammonia channel. The human RhCG ammonia transporter was found to have a similar ammonia-conducting channel structure. It was proposed that the erythrocyte RH complex is a heterotrimer of RhAG, RhD, and RhCE subunits, in which RhD and RhCE might play roles in anchoring the ammonia-conducting RhAG subunit to the cytoskeleton. Based on reconstitution experiments, purified RhCG subunits alone can function to transport ammonia. RhCG is required for normal acid excretion by the mouse kidney and epididymis. Structure. The structure of the ammonia channel from E. coli was, at the time of its publication, the highest resolution structure of any integral membrane protein. It shows a trimer of subunits, each made up of 11 transmembrane segments TMS and containing a pseudo-two-fold symmetry. Each monomer contains a hydrophobic ammonia-conducting channel. While prokaryotic ammonia channel proteins have an N-terminal region, which acts as a signal sequence and is cleaved in the mature protein, the Rhesus glycoproteins retain this as a 12th transmembrane helix in the mature protein. Substrate specificity. Most functionally characterized members of the family are ammonium uptake transporters. Some, but not other AmtB proteins, also transport methylammonium. Detailed phylogenetic analyses of plant homologs have been published. In E. coli, an H4+, rather than an H3, may be the substrate of AmtB, but controversy still exists. If an H4+ is transported, K+ possibly serves as a counter ion in an antiport process with K+, and that one histidine removes a proton off of an H4+ to yield an H3. Transport reaction. The generalized transport reaction catalyzed by members of the AmtB family are suggested to be an H4+ out equals an H4+ in. Mechanism. The X-ray structures have revealed that the pore of the AmtB and RH proteins is characterized by a hydrophobic portion about 12 along, in which electronic density was observed in the crystallographic study of AmtB from Escherichia coli. This electronic density was initially only observed when crystals were grown in the presence of ammonium, and was thus attributed to ammonia molecules. The AmtB/RH protein mechanism might involve the single-file diffusion of NH3 molecules. However, the pore could also be filled with water molecules. The possible presence of water molecules in the pore lumen calls for a reassessment of the notion that AmtB/RH proteins work as plain NH3 channels. Indeed, functional experiments on plant ammonium transporters and RH proteins suggest a variety of permeation mechanisms, including the passive diffusion of NH3, the antiport of an H4+/H+, the transport of an H4+, or the cotransport of an H3/H+. Lamerux et al. Discuss these mechanisms in light of functional and simulation studies on the AmtB transporter. Regulation. In E. coli, the AmtB gene is expressed only under limiting nitrogen levels to yield the AmtB protein. It is co-expressed with the GLNK gene, which encodes a PII protein. This protein is also trimeric and remains in the cytoplasm. It is covalently modified by a U/U uridylated group at a 51. The hydrolyzed product, adenosine 5-diphosphate, orients the surface of GLNK for AmtB blockade. When nitrogen levels outside the cell rise, the ammonia channel must be deactivated to prevent excessive ammonia entering the cell where ammonia would be combined with glutamate to make glutamine, utilizing ATP and thereby depleting the cell's ATP reserves. This deactivation is achieved by deuteridylation of the GLNK protein, which then binds to the cytoplasmic face of AmtB and inserts a loop into the ammonia-conducting pore. At the tip of this loop is an arginine residue which sterically blocks the channel. Human ammonia transporter-related proteins RhAG, RhBG, RhCE, RhCG, RhD.